Welcome back, everybody, to Chapter 9, the biologically important amino acid derivatives. So in this chapter, look at our learning objectives. We're going to describe the importance of the following special products that are derived from amino acids, their biosynthetic pathways, and special functions. We're going to talk about heme. When we talk about heme, we talk about hemoglobin, myoglobin, those kind of things. And we've mentioned that a little bit here and there. It's kind of our oxygen transportation pathway. That's what we use heme for. Creatine and creatine phosphate. This is going to be dealing with kind of our initial energy sources. We can convert these to ATP in the muscles. Purines and pyrimidines, those are DNA stuff. Or hormones like thyroid hormones and melatonin. Or neurotransmitters like our catecholamines, serotonin, and nitric oxide being a vasodilator. Pigments, melanin, big important thing here, differentiating melatonin from melanin. They sound similar. It can be easy to confuse them when you're rushing on a test or a quiz. Just to make sure you read that carefully. And also histamine. Histamine we've mentioned here and there a couple of times. So we're going to dive kind of how we actually create some of these specific things. Again, like the other videos, I'm going to try and cap this one at about half an hour. We'll see how far we get through in the first video and make it into a couple different parts as we go through all of this. So also kind of to start with, the things that we're really going to look at. A lot of this is going to be kind of, hey, draw these out, learn some of the intermediates, and really understand the function of each of these, as well as what the enzymes, specifically those rate-limiting step enzymes. We want to look at the intermediates, the rate-limiting enzymes, and then the function or the purpose of these. Okay, that's what we're really going to be focusing on as we read through this. So amino acids are important precursors for many physiologically important nitrogenous compounds, such as hemoglobin, the nitrogenous bases of nucleic acids, like those purines and pyrimidines, certain hormones, biogenic amine neurotransmitters, again, talking about amine, talking about that nitrogen, vasodilators, and gaseous neurotransmitter like nitric oxide, as well as some peptides. So we're going to go over just briefly kind of what each of these are and then jump into the actual formation process. So heme forms the core of hemoglobin as well as myoglobin molecules. Remember, hemoglobin is the one that is found in the blood. Myoglobin is the one that is found in the muscle, and its function is to carry oxygen and get oxygen around, so they're the oxygen carrier proteins in our bodies. They also are responsible for carrying out the carbon dioxide, so while we do dissolve some of it directly into the blood, our hemoglobin, myoglobin stuff will bind the carbon dioxide as well in order to get it back to the lungs so that we can breathe it out. Heme is also present in the center of cytochrome molecules that functions in the electron transport chain system. Remember the cytochromes in the electron transport chain. When we were learning all about that, we had those different, so if I can get this to give us a good picture. Remember all those different cytochromes, cytochrome C, and we had a bunch of different components within the electron transport chain that was responsible for passing along those electrons and helping us to create ATP for our muscles. The heme moiety, remember moiety just means kind of a part of, I believe I wrote the definition down here someplace. So a moiety is a fragment of a molecule that is part of its chemical structure and can include a substructure such as a functional group. So a moiety is just the different components that all get put together to create something bigger. Or moiety can just mean that it's going to be like the precursor of something else, either because things get added, things get taken away, etc. And that can be further metabolized to form biliverdin and bilirubin. We'll talk more about biliverdin and bilirubin. These guys right here are going to be very, very, very important, especially bilirubin. When we talk in the future about kind of liver, gallbladder health, kidney kind of stuff, as well as a lot of our lab tests when we talk about bilirubin. And we'll get into exactly what that is. Part of what you'll be required to do on this upcoming test for this class is identify and talk about, we'll talk about conjugated versus unconjugated bilirubin and kind of what that means. So we'll talk about that a little bit more when we get a little further down. Just wanted to introduce that idea to you guys. Creatine or creatine phosphate. I've also heard it be called creatine, creatinine. Most of them you're going to see it creatine. There's another chemical called creatinine, which is something slightly different, but kind of is related to, and we'll see. I'm not sure if it's in this chapter that we talk about that, or if it's going to be in future courses when we talk more about like kidneys, like in Phys 2. Also known as phosphocreatine. It is used as a reservoir of high-energy phosphate bonds. The skeletal muscle converts phosphocreatine to produce ATP and uses ATPs during muscle contraction. This is going to be, using creatine is going to be the first couple seconds. Let me double check exactly what the time is on that. So our creatine phosphate, also known as phosphocreatine, can provide energy for muscles for 8 to 15 seconds during short, intense exercise. It's called the ATP-PC, so ATP phosphocreatine system, and it's most active in athletes who perform explosive movements like powerlifting or a 50-meter dash. So kind of when you start to combine some of these thoughts and these ideas, when we think about muscle typing, remember way back, actually bringing in kind of Phys 1 here, we talk about our type 2A and B muscle fibers. Type 2B muscle fibers are our fast glycolytic muscle fibers. They produce a lot of power, but and they fatigue rather quickly. So our creatine phosphate system kind of will act with those type 2 muscle fibers. Purines and pyrimidines are important molecules that synthesize the nucleic acids, DNA and RNA. Our hormones are synthesized from amino acids such as thyroid hormones, thyroxine, and triiodothyronine are produced from tyrosine. These are going to be our T4 and T3. They're from tyrosine. Tryptophan is a precursor for the synthesis of melatonin. This is a hormone produced within the pineal gland, which regulate our circadian rhythm, the light-dark cycle, or the day-night cycle. So these are ones that we're really going to want to remember. We're really going to want to remember tryptophan, and we're really going to want to pay attention to tyrosine. These guys here are going to be important for your exam in this course, but these are also heavily tested on boards, kind of the process of everything that can come from tyrosine and tryptophan. Boards really, really likes those questions which are coming up, 2-3 tries from now is when you guys will start taking your first board exam, and that's going to be involved in that. So lock that away into long-term memory. That is going to come back. Melanin is the dark-colored pigment in our skin. It's synthesized also from tyrosine within melanocytes, which are in the skin. The pigment prevents our skin from damage causing ultraviolet lights. Neurotransmitters, some neurotransmitters such as catecholamines, dopamine, norepinephrine, and epinephrine are synthesized again from tyrosine. Tyrosine does a whole bunch of stuff as their precursor molecule. The neurotransmitter serotonin is synthesized from the amino acid tryptophan. There's tryptophan again. The amino acid arginine also produces a short-lived chemical messenger molecule, nitric oxide. Nitric oxide, if you have heard of that before, it's because that's something that they will give to people who are experiencing episodes like heart attacks or episodes of like severe high blood pressure. They'll give them nitric oxide. It helps. It's a vasodilator to help all the blood vessels dilate and to lower blood pressure and to help blood flow through the vessels. Immunotransmitters such as histamine is synthesized from the amino acid histidine. Histamine is important in many of the inflammatory reactions. Again, remembering that histamine is a vasodilator because when we have inflammation, it's an inflammatory. It's an inflammatory immunotransmitter, and so it's going to cause vasodilation. Histamine is a little bit weird in the sense that while it dilates the blood vessels, it constricts the airways. So we always remember histidine or histamine as a vasodilator, bronchoconstrictor. That will come back up a lot in the future, so just keep that in mind. So starting with glycine for heme, creatine, and purines. So it contributes both carbon and nitrogen to form the heme, creatine, and the purine bases. Glycine is also important as a source of 1-carbon moiety for the folate coenzyme system for the biosynthesis of glutathione and glycine conjugated bile acid, glycocholic acid. And we talked about that a little bit before when we talked about the liver and the bile salts, all that kind of stuff. So glycine also reacts with a number of compounds to form conjugates by increasing their water solubility and thus aiding in their excretion in the urine. So conjugates, we'll talk about that a little bit later when we talk about like that bilirubin and stuff. So heme biosynthesis. So heme is an iron-containing prosthetic group. That's super important. Remember, iron is what is inside of heme. It is required for the synthesis of hemoglobin, myoglobin, and cytochromes. This is the beautiful structure of heme with iron there at its very center. We talk about these are all nitrogenous, so there are our nitrogen bases there. And so heme consists of what's called a porphyrin ring, or built around an atom of iron. And actually, you'll see iron, a couple different names for iron. Like if you ever see the word ferritin, so it was another one. If the other one comes to my mind, I'll let you guys know. But ferritin, if you ever see something like this, ferritin is kind of that storage form of iron. So ferritin, fyrin, kind of that pH, that F sound, a lot of times is going to be indicative of iron. So we have porphyrin ring, so this whole ring structure built around the iron. And the way that we get that is we have these 4 pyrrole rings. Okay, the 4 pyrrole rings. If we look here, ring, 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 ring. You can see those. Each of these rings are joined together by methynyl bridges and the nitrogenous ring members surrounding the Fe. So 8 side chains are attached to the pyrrole rings, 2 on each pyrrole. So this is going to be kind of where some of our questions are going to come from. Understanding, so we have pyrrole rings. We combine those together to form a porphyrin ring, and then we also add on 8 side chains as well. You can see these side chains here. Side chain, side chain, side chain. All of these guys are our side chains. And that's the production kind of heme. This process of heme synthesis occurs in erythroid cells as well as the liver. Whenever we think blood, there are 3 places I want you guys to really think about. I want you to think about the liver. I want you to think about bone marrow, especially in the long bones and the flat bones. So bones like the femur, the humerus. Think about the pelvis. Those kind of big bones are where we do a lot of our making of bone marrow. It's important to remember that part now, because in the future when you start talking about diseases and conditions, if you haven't already in your clinical imaging courses, a very, very common condition that you'll talk about in the elderly population is called multiple myeloma, and it's a primary tumor of bone, and it's a blood-based tumor. So it's due to a malproduction of blood cells that kind of tumor comes around. So we see that often in the femur, in the pelvis, a lot of times in the skull, some of those big flat bones, stuff, and those ones that are producing a lot of our blood. And then of course the liver detoxifies, it cleans a lot of stuff out. A lot of blood runs through the liver. And the other one I want you to think about is the spleen. The spleen is really responsible for a lot of cleaning out and breaking down some of the yucky blood cells, so the old red blood cells. And so you'll talk more about the spleen and kind of its function in future courses. So heme synthesis starts in the mitochondria with the condensation of glycine and succinyl-CoA. Succinyl-CoA, that's one of our intermediates of the Krebs cycle, forming delta-aminolevulinic acid, also known as delta-ALA, which is catalyzed by that delta-ALA synthase. Remember, synthase means that we're synthesizing, we're making something, and it uses pyridoxal phosphate. Before you read ahead, what B vitamin is that? Pyridoxal phosphate is the active form of B6. So again, think about our B6 vitamins. B6 does 2 different things. It does transamination as well as amino acid metabolism. So right here, amino acid metabolism. Transamination is like the passing of different functional groups onto something else. And that is required for the production of heme. So this specific enzyme is the rate-limiting enzyme and controls heme biosynthesis by feedback inhibition. So right there, immediately, this enzyme right here is one that you absolutely have to remember. Delta ALA synthase. Make sure you remember that one, because that's going to be one that's going to focus on test questions, because that's going to be the rate-limiting step. So within the mitochondria, that succinyl-CoA combines with the glycine using the delta-ALA synthase, as well as the pyridoxal phosphate, or B6. We get the aminolevulinic acid. From there, we are actually going to create 2 of them. We're going to join 2 of them together, and then we get a dehydratase enzyme. Dehydratase, dehydrogen, we're taking away water. And then we get a porphobilinogen, which is also known as pyrrole, which we saw earlier. From here, we're actually going to go into the cytosol. Before, we're going to just read through this a little bit, just make sure we don't miss anything. So the alpha carbon and the alpha nitrogen atom of the glycine are used to synthesize the porphyrin moiety of heme. Remember, moiety, we're just talking about a precursor. We're talking about one component, one part that's all going to kind of come together to form something bigger or something different. And remember that alpha carbon, alpha nitrogen, we're just talking about here how we have our carbons, we have our nitrogens and stuff within the amino acid that we're going to be using in this process. Remember, because glycine is one of our amino acids that we're going to be using. Delta ALA diffuses to cytosol, where 2 molecules of the delta ALA further condense to generate porphobilinogen. This reaction is catalyzed by the delta aminolevulinic acid dehydratase. Again, one of the enzymes we'll want to remember. I recommend this whole process. What I would do is get out a piece of paper now, and as we go through this chapter, write down each of these steps with specifically just focusing on the intermediates. It's like succinyl-CoA plus glycine, arrow, that delta ALA synthase, and PLP combine, all get used to create the delta ALA. And give yourself like a nice little cheat sheet study guide just based off of getting rid of, all of like the crazy drawings and stuff. It's just that it's a little bit easier to look at. It gives yourself a nice way to study these. Taking a little step back to look at clinical correlates. So lead poisoning inactivates the delta ALL dehydratase, leading to anemia. Anemia meaning low red blood cells. We don't have the type of red blood cells, enough red blood cells that we need. Because if we're not producing heme is going to be one of the main base components in order to produce red blood cells. So if we don't have the heme, we can't make new red blood cells. Another note, red blood cells will be called erythrocytes. So remember that. Erythrocytes, if you see that, that just means red blood cell. 4 molecules of porphobilinogen condense to yield. Sorry, step back. We're kind of jumping. This is just like an extra little tidbit right here. So we're jumping from there, continuing. Have 4 molecules of porphobilinogen condense to yield porphyrin uroporphyrinogen 3. Then there's a decarboxylation reaction which produces coproporphyrinogen 3, which diffuses into the mitochondrion and finally convert to heme. And iron is incorporated into the protoporphyrin ring in a reaction catalyzed by ferrochelatase or heme synthase. So this is just an aka heme synthase to form the complete heme molecule. A lot of these kind of little extra guys, if you have the time and the patience, to practice this a couple of times and go through it, great, learn all of them. If not, the ones that I would focus on, know that we're starting with glycine and succinyl-CoA, that we go to ALA, and that we go to the porphobilinogen. All of this happens, this first chunk right here, happens in the mitochondria. And then the next chunk of this part, remember that this happens right here. All of these guys happens in the cytosol. And then finally at the end, we go back into the mitochondria. Okay? So I'd remember these first 3, important to remember. Some of these in-between steps, not so much. But you'll definitely want to remember ferrochelatase, and then the synthase, and the dehydratase. If you can remember all of them, better. but especially those ones that I just pointed out. Let me actually just highlight them so it's easier. So this, we want to make sure we remember this. All of this. Again, like I said, ideally all of it, but if not all of it, remember these specifically for sure. Again, heme is complexed with proteins to form hemoglobin, myoglobin, and cytochromes. So in other words, we take our heme and then we jam it in with other stuff, with other proteins. Remember we talked about how hemoglobin is a quaternary protein. So essentially we're going to be taking a couple different components of heme and some other stuff, jamming it all together in order to form hemoglobin, myoglobin, and the cytochromes. Now the degradation or the breakdown of heme. So heme is degraded to something called bilirubin, which can then be conjugated with glucuronic acid and excreted in the bile. So we'll get through this part and then start the next part. So bilirubin itself is I'll make sure we describe it in a simple way. Bilirubin is essentially a leftover product from breaking down heme. Right? Anything with heme, most of the time we're talking about hemoglobin, aka red blood cells. So when we break down red blood cells, we get an extra that's called bilirubin. This bilirubin is then in the blood, and as soon as we break it down, it is unconjugated. So red blood cells get broken down, and that releases bilirubin, specifically unconjugated form. All that means is that specific bilirubin hasn't undergone a specific chemical reaction that it needs to undergo in order to then, be used to do other things within the body. Where it goes to get conjugated or to get acted on by glucuronic acid is going to be in the liver. So bilirubin gets conjugated in the liver, then excreted in the bile. Because remember that the liver, within the liver, we're kind of producing the bile, and it's going down and it's going into the gallbladder and then held in the gallbladder, which then releases it into the small intestine in order to digest stuff. So what's really cool is, and we don't talk about this a ton in this course, but we touch on it, is how can we tell, or how can this tell us where we have an issue, right? Sorry, give me one moment. Okay, so one of the ways that we figure out kind of what's going on, especially, is that we actually take blood draws. So we draw blood from a person and we look at the bilirubin, and we can break it down and look and see, okay, what are the levels of unconjugated versus conjugated bilirubin within the body. And so if we look at it and we see, oh, there's a ton, so high levels of unconjugated bilirubin in the blood, we stop and we think, okay, if there's high levels of unconjugated bilirubin, what's going wrong? Okay, what organ is having trouble that would prevent this from being conjugated? That tells us that there's an issue with the liver, that there's a liver issue. If we have high conjugated bilirubin, that means that it made it to the liver acted on it, it did its thing, but then something after the liver is having a lot of issues. And so that we tend to lean towards gallbladder issues. Liver issue, especially something like cirrhosis of the liver. Cirrhosis just means like breakdown, scar. It literally means scarring of the liver. So stuff like alcoholism, that kind of stuff, can really damage the liver, where then we would have high unconjugated bilirubin. The gallbladder, we can have gallstones. The technical name for that is, let's see if I can spell it, cholelithiasis. There we go. Gallstones. Essentially, the gallstones, what they do is they go and they plug up. They're like these little fatty balls, kind of like kidney stones, and they go and they block the duct where the bile is supposed to go down through. And because of that, the bile gets backed up and kind of backflows, and we get more of the conjugated bilirubin in the bloodstream, which then we can see on our lab values. Okay, this is the actual process breaking down of heme. So this is, one step back. This is what we're going to want to remember. Unconjugated versus conjugated bilirubin, where it gets conjugated, and what's kind of going on if it's not. Another reason why we can have really high conjugated bilirubin is if we have a blood issue where we are breaking down a ton of red blood cells. This would be something where, like, our body's recognizing that our red blood cells that we have are not the way that they're supposed to be. So you'd also see this with somebody kind of like with like sickle cell anemia, where, you know, they have red blood cells that aren't supposed to be that way, or some types of blood cancer, where suddenly a lot of your red blood cells are having to be destroyed because they're not working properly. Okay, so those are the kind of stuff that you'll see with those conditions. You won't get a ton of this clinical side of things in this course. We'll have a handful of questions about this kind of stuff, but you'll go way more in depth in the future with this. Okay, so starting with heme, we get heme oxygenase. We pull out this carbon monoxide, pull out the iron, and once the iron has been pulled out, we get biliverdin. Then we have a biliverdin reductase. And then we have the bilirubin, and the bilirubin is in the blood. So it can travel to the liver. It can go into the intestine, where it's in the bile, and from bilirubin, it'll be converted to urobilinogen, urobilinogen can either be transported to the kidney and be excreted as urobilin, or it can continue on to be stercobilin, which, if my memory serves me, the questions he's going to ask you, more so than like these specific enzymes and stuff, he's going to be heme going to biliverdin, biliverdin going to bilirubin, and then kind of where does bilirubin go to? It goes to the liver, goes to the bile, and then can also be excreted. Here's a last little information, and we'll stop after this point. So when red blood cells reach the end of their useful lifespan, which is 120 days, a lot of teachers like this question. 120 days is how long red blood cells typically last. They are phagocytosed by cells of the reticuloendothelial system. So just phagocytosed, they're swallowed and they're broken up, they're broken down. Globin is cleaved to its constituent amino acids. Iron is returned to the body's iron stores. Heme is oxidized and yields carbon monoxide and biliverdin. Biliverdin is then reduced to bilirubin, which is transported to the liver bound to serum albumin. So it's bound to the serum albumin. Again, if we look at this and we think, if there's a ton of bilirubin in the body and it's unconjugated, this is part of the big reason. When we have a liver issue, like liver cirrhosis, scarring of the liver isn't working properly. The liver is responsible for producing our albumin. So if we're not producing albumin, then the bilirubin can't bind to it as much in the blood and continue throughout the whole body and get back to the liver in order to be conjugated. So in the liver, bilirubin reacts with UDP glucuronate to produce a more water-soluble compound, bilirubin monoglucuronide. That bilirubin monoglucuronide is then further conjugated with another UDP glucuronate to form bilirubin diglucuronide. In the intestines, the bacteria deconjugate that bilirubin diglucuronide and convert the bilirubin to urobilinogen. Urobilinogen is oxidatively degraded by microbes to stercobilin, the pigment that gives feces its brown color. Where you're going to see test questions in all of this chunk, again, is heme to biliverdin to bilirubin. They'll probably ask about the serum albumin, so how biliverdin, or I should say how bilirubin is transported to the liver thanks to serum albumin. What it reacts with, so bilirubin reacts with the UDP glucuronate. And then you're also going to be asked specifically this last bullet point down here. This is a question that a lot of people like to ask, that urobilinogen is degraded by microbes in the intestines, specifically the bacteria, the microbes of the intestines, degrade the urobilinogen in order to give feces its brown color. Okay, we went a little bit over in this one. We'll continue in the next part with creatine.